Hey, so today I'm going to be presenting my team target with the Unipro ID of U2RC82. So the first slide here, we have pairwise alignment and percent identity. And as you can see, we have a calculated percent identity of 68.48% to the project standards. So it's a little farther. Um, we perform the pairwise alignment and obtain a percent identity um, for our protein U2RC82. And it's a PepCK protein. From our results, we see that critical amino acids are the same between the standard and target, which means that like key characteristics are preserved, but it was still somewhat farther. On the next slide, um, this is our phylogenetic tree, and our target, you can see that the evolutionary distance is 0.3814, which is not super close, which is under 0.25, but it's not super far above 1. And there's a moderate distance, so that there'll be some characters that's preserved, but it still has phenotypic differences. So this is our Gibson assembly. And in Gibson assembly, we created two PCR sets, one constructing the linearized plasmid PCR product, and the other being our target PCR product. And it contains overhang to generate the plasmid through Gibson assembly. And this is how we made and reproduced and amplified our protein. And this is a template right here. Okay. Next slide, we have biophysical protein characteristics. So we determined that the graph on the left, you see the processing tag was inserted into our plasmid through the target PCR for later purification using the IMAC. And on the right, it shows the unprocessed recombinant protein amino acid sequence of our target. Yeah, so here's our number, our molecular weight of the unprocessed. He has a process, which is lower as expected in theoretic PI. So recombinant protein expression in bacteria. We use IPTG addition in order to bind the lac repressor. And this prevents it from blocking the lac promoter. With the lac repressor block, the lac promoter can translate the T7 RNA polymerase that then binds to the T7 promoter, which then translates our target gene to the target protein. And IPTG addition basically produces more target protein, and this is how it's done. For the immunodetection scheme, these are the steps basically we did for immunodetection, and we separated protein through Western blotting, transferring the protein to membrane, and then subsequent staining and chemiluminescent imaging are used of the protein to visualize and detect it for the flag tag. And the gluconeogenic activity, we produce a gluconeogenic assay of the NADH oxidation. So this basically equated the loss of NADH to PEP formation of where this is our assay reaction over here. And then we measure the amount of NADH through absorbance. So our PCR results and determination. So we did this in the beginning, and we identified our target by amplifying our DNA and binding it to primers, our specific for certain target. And through this, we eventually identified our target to be the U2 RCA2, which is primer set 2. So for the calculation of transformation efficiencies, we calculated the transformation efficiency using DH5 alpha E. coli cells. And these cells are competent to take up the plasmid we created using Gibson assembly. And then we then plated at different concentrations in order to evaluate the transformation efficiency. And you can see at the different concentrations and efficiency depending on it. And on average, it was around 5 for the control. And then ours was around a little lower on 2.43. For the DNA sequence and amino acid translation, basically we have just determined DNA sequence and translated amino acid sequence over here on the right, and then DNA sequence on the left. And so for the validation of the team target DNA sequence, we validated our target through sequencing validation through BLAST of both the nucleotide sequences of our DNA sequence. And BLAST N, the identity is 97, and then BLAST X is 100%. the DNA purity spectrum and yield estimate, we obtain DNA purity spectrum and the yield estimate through measurements of absorbance of the plasmid DNA solution with a concluding concentration of 47.2 nanograms per microliter. The purity was analyzed on the following values you see on the right over here. And this at the different absorbance levels. For the optical density and growth curve in IVTG induction, this shows the IVTG addition point, it's highlighted in red, as you can see. And we know that the OD 600 value raised greatly and basically showing that our target plasmid was replicating really rapidly. So our first method of purification using the IMAC, and by having the metal bind to the histine of the processing tag, and it causes them to bind closer to the rest. As you can see here, the different letters correspond to different steps. Where A, you have the addition crew lysate, and then the wash buffer at B, and then the elution buffer, and at the end, as you can see, the purified coming out of the column. And then we perform size exclusion chromatography, where we use imidazole, and that removes the target protein from the solution, having it move slower through the beads because it's smaller, and then the target proteins that are larger, so it filters out larger molecules, and while binding the target with imidazole, yeah. For the batch processing, after our purification, we quantified the amount of loss and found that we did not lose much of our product. So it's processing loss about 3.10 milligrams, and our bound protein loss is 2.6 milligrams. But our batch processing went smoothly, and bound protein loss was similar to the processing loss. We then determine the yield by calculating concentrations and then multiply by volume. And through this formula is the Spear-Lambert equation, the Lambert equation. And you can see our final yield was 1.80 milligrams per milliliter times 9, and it gave us value of 16.2 milliliters. And then, yeah, so that was our final. And then our mass spec, we performed the spectroscopy analysis in order to determine the molecular weight of our protein. It only found a 1.11 Dalton difference between the mass spectroscopy and the predicted molecular weight. And so this is really close. And basically over here, you see a large peak giving us a value of the mass for our protein. And through the param results back in the earlier units, we see that this is really similar. For the Western blot analysis, stain gel image is on the left, and this reflects the staining lanes listed on the right, and the stain blot verified that our protein got transferred to the membrane for immunodetection, which is this one. And then we see that in lane 5, it should contain the flag tag, while in lane 9, in the chemiluminescent image, is absent because it does not contain the flag tag. So basically it means that there's no processing tag. And then here are the lanes, it's different contents and mixtures to give us an idea of how fast it should be moving through. As you can see at the end, it became thinner, and there was not a lot of different sizes through the gel. So the protein structural alignment for the project standard, we use AlphaFold to try to determine the hypothetical structure. And the protein folding prediction is seen on the left. And you can see the differences between the standard and target are roughly 1.96. And as closer to 1 is the higher the score is better. And then TM should be closer to 1. So it's pretty high. And then the Ramcharan plot shows the position in the alpha helix and the beta barrels of the protein. And you can see the low probability score of 0.82. And we want that lower. And so for our prediction from the modeling analysis, we got our PLDT, DDT score of 95.63. Our target was 94. The project standard is 94 and the target is 95. So it's roughly pretty close. And it's it's really similar to the project standard, the target. So we expect something similar in terms of certain characteristics of the protein, but then we predict that there'll be a 15 difference in TM and negative 21 difference in specific activity. So the melt temperature, as you can see here, um, is basically where half the protein unfolds and then it gives off and is measured through absorbance values of the fluorescence. Like the more protein that starts to unfold, the more um, fluorescence it gives off. And basically, you see here, we did have got an average value of 43.3 and the standard deviation is 0.3. So it's in all of our trials, it's pretty close around 43. So we're pretty confident in this value. And then it's a little lower than a bit lower than the product standard, which is in these wells, we took derivative and then found the minima to see the maximum fluorescence to know the TM. And then the lower kind of means it's a little more unstable. And for the gluconeogenic assay, we basically did it to identify if it was a PEP-CK. And as you can see, that's with no manganese, the activity was lower, and then with manganese, you can see it was higher. So with manganese, it gave us a higher specific activity, which gave a lower PEP concentration over here. Okay, so this is our reproducibility results where we use another U2 or CA2, and we got an average temperature of TM of 45, product standard is 54.5. So it's pretty accurate, pretty similar, and our specific activity is roughly around the same as well. It's a little lower than the project standard, and the average temperature is about the same. This is our final data set. We have some of these values. These are the temperatures at the different units that we measured versus the project standard, and these are specific activity values. And our protein did display magnesium dependent PEP-CK activity, which is what we wanted to see for characteristics of PEP-CK. And DNA was verified, and it has a percentage 68.48%. All right. Yeah, thank you.